Hi all, Dr. Clark here, and we're going to finish off photosynthesis by talking about the Calvin cycle, or what sometimes is called the light independent cycle. So we got done with last time talking about the light dependent cycle, so photosystem 2, photosystem 1, the products of those being ATP, NADPH. Now we're going to talk about the Calvin cycle and how that plays a role in the main role in the construction of carbohydrates. The whole, I guess, purpose or premise behind photosynthesis, that's what we're going to talk about now, is that building of carbohydrates. This is how it's done, and it looks really complex, with Rubisco and RUBP and carbon dioxide coming in and making phosphoglycerate and all these intermediates, and it is kind of daunting and a little bit complex, but we're going to work through it. We're going to talk about each kind of step along the Calvin cycle and what's going on, how much energy is expended, and whatnot in this lecture. Okay. First, before we do that, though, I want to plug two books, and one that I'm a really big fan of, and that's Biomimicry: Innovations Inspired by Nature. Okay. And so I talked about this before, about like photosynthetic cells, about hydrocars and things like that. And I just figured some of you might want to check out some books on these ideas. And where did the idea for a solar panel come from? Well, it came from nature. Where did the idea of creating an engine that runs on water, where did it come from? Well, it came from nature. And so the biomimicry book here is excellent. This one's good too. I mean, just in my opinion, it's not as good as this one. But again, these are inventions that were inspired by nature. And so in some cases, it's, I don't know, there's kind of a more loose uh, connection compared to the biomimicry innovations inspired by nature. But both are excellent books. If you can pick one up, or if you see one in the library, grab it, read it. You really won't be disappointed. It'll point to a lot of things that will just make you think, like, well, what else is there to know? Like, I mean, for example, how many drugs, how many antibiotics that we have today were created by or our knowledge from things that are created in plants? Pretty much all of them. And so that's really where we're going with medicine even today, is developing new antibiotics and new drugs and things like that. It comes from plants mainly, but sometimes from fungi and sometimes from bacteria and things like that. And that really is where we get a lot of our antibiotics. Well, it's also where we get a lot of our innovation and inventions really come from nature also. So check these books out if you like, and I don't think you'll be disappointed. All right, so moving into how to build new molecules. Okay, and so we just got done talking about the light dependent reaction. We talked about the two products from that, ATP and NADPH. So now we're going to take those ATP and NADPH molecules. So ATP is utilized for inorganic reactions. We talked about this before. NADPH is needed whenever an electron is needed or another proton is needed. And so we say that it has reducing power. So it means that it can donate an electron to another molecule and be oxidized. Right? And so the Calvin cycle is really just the construction of new molecules from simpler molecules, and utilizing energy from the light-dependent reaction. Okay? And again, it can be either called light-independent reaction or the Calvin cycle. So, those terms are used interchangeably often. And so you might see on an exam or questions on a quiz or something like that, Calvin cycle or light-independent reaction. It's just wording. Same thing. Okay? Now, when this is completed and you go through the light-dependent reaction and the light-independent reaction to construct new molecules, it's considered C3 photosynthesis. Okay? At least the straight method of going through light-dependent during the day when photons are available, light-independent at night or during the day when photons are available, that's the C3 photosynthesis. So when you hear things like, oh, that's a C3 plant, that means it's doing photosynthesis the C3 way. There are also what we call C4 plants that do photosynthesis a little bit different, and we'll talk about them a little bit later on in this lecture. And there's also what we call CAM plants, which do photosynthesis a little bit different also. And we'll talk about both those types in this lecture just later on. All right, so the Calvin cycle happens in 3 main stages. So remember, anytime it says cycle, then that means it's circular, and that means that most often whatever's utilized at the beginning returns to the cycle, and so it's reused. Most of these cycles in biology are basically just like a recycle center. Okay? You take it there, it gets destructed and reconstructed, okay? And it just happens over and over again. So the Calvin cycle is no different. The first part of the Calvin cycle is carbon fixation. So carbon dioxide from the atmosphere is brought into the plant, okay, through the stomata, which we talked about before, these little openings at the bottom of the leaf. So the carbon dioxide is going to be brought in, okay, and then that carbon dioxide is going to be attached to a molecule that we call RUBP, or sometimes we just call it RuBP. And so that molecule is a 5-carbon molecule, and when carbon is fixed to it, this molecule becomes a 6-carbon molecule. We'll come back to this. Don't worry if you don't get it all right now. We'll go through this many times. Okay. So then, through the process of shuffling carbons around and breaking bonds and reforming bonds and adding phosphates to the molecule and whatnot, sugars are made. Okay? Particularly, glucose is made. Okay? Then the remaining RUBP and the remaining 5 carbons that are left after the sugar is made, okay, goes back and you get a little more shuffling going on and you reform the original molecule RUBP, and then you can bring in more carbon dioxide from the atmosphere and do it over again. Okay? And so this is really the process by which glucose is formed. And we're going to go through each one of these in much greater detail. Okay? So the first part, you have 3 molecules of carbon dioxide that's going to come in. Okay? And you have 3 RUBP molecules. And through the combining of that carbon dioxide to that 5-carbon RUBP, you get what we call 6,3-phosphoglycerate, okay? Which means that it's been phosphorylated. That means that a phosphorus has been added to this. Okay? Now, that 6,3-phosphoglycerate is going to go through changes. So the carbon's going to be moved, more phosphates are going to be moved, okay? And more phosphorus is going to be added. That's where ATP comes in. Okay? ATP is going to phosphorylate this molecule. Okay? So it's going to supply energy to this molecule to break bonds, reform bonds, etc. NADPH is going to add electrons needed. Okay? And so what you're going to have is you're going to create 6 molecules of glyceraldehyde 3-phosphate. That is the building block. glyceraldehyde 3-phosphate, is the building block of glucose. If you combine glyceraldehyde 3-phosphate together, you will get glucose. Okay? And so one molecule of these 6 glyceraldehyde 3-phosphates, one molecule is going to be donated to the formation of glucose. The remaining 5 molecules will go through more transformation by adding more energy. You're going to remove phosphates, you're going to change this molecule, and you're going to add more of these. So you're going to break a couple of these glyceraldehyde 3-phosphates down and combine and get your beginning starting material. 3 molecules of RUBP. So we'll look at this and we'll go through this again, but this is how glucose is formed. Okay? This is the Calvin cycle. So again, let's just kind of reiterate what we just talked about. Now, one molecule of glyceraldehyde 3-phosphate is added to form that glucose molecule. So if you're thinking this through and you're trying to decipher, well, how much is needed to form one glucose molecule? Well, only one carbon can be added of carbon dioxide. Only one carbon from carbon dioxide can be added per turn of the Calvin cycle. And if you remember, the chemical equation for glucose is C6H12O6. So if you can only add one carbon each turn of the Calvin cycle, then you know that the Calvin cycle must turn 6 times in order to form a single glucose molecule. Okay. Now, the nice thing about the Calvin cycle is that it recycles the material that it started with. So yes, I mean, it's kind of inefficient in the fact that you have to turn it 6 times. You have to take in carbon dioxide, turn it, switch things. You got to burn energy to form, glyceraldehyde 3-phosphate, and you got to do that 6 times. Okay? That's not super efficient. However, RUBP is always going to return to the cycle. Okay? And so you're not really breaking anything apart too terribly bad. And you're just taking carbon dioxide from the atmosphere. Now, the issue that we have often is that it's taking energy. It's taking ATP and NADPH, okay? 7 molecules most of the time of ATP, somewhere in there. And that's coming from that light-dependent reaction, and that's going to be donated to the system. So the great thing about photosynthesis is the energy to get ATP is free if it's coming from the sun in the form of photons. It's free. So even though this might not be the most efficient system to make glucose, many people, engineers, could probably design a different way for this to occur, but this is the way that evolution designed the system. And yes, it seems like a waste of energy to form a glucose molecule, but nonetheless, it's a cycle, and it keeps recycling the reactants over and over again. The other piece is after ATP after a phosphate has been removed from ATP, ADP goes back in to the light dependent cycle. After you remove an electron from NADPH, NADP moves back into the light dependent reaction. So these reactants are reusable. Okay? And so just what I said right there. So after you steal a phosphate from ATP, it becomes adenosine diphosphate, okay? And it's available for chemiosmosis in photosystem 2. After you take the electrons away from NADP, it goes back into photosystem 1. Okay, so these are recycled reactants, and it just can keep carrying on as long as there's photons available and water available. This process can continue, because remember, you got to get the electrons from breaking the water molecules. So as long as photons and water are available, this can occur over and over and over again, and the plant can make new sugars as long as carbon dioxide is coming in. So here, let's look at it again. 3 molecules carbon dioxide comes in. You have 3 molecules of RUBP. Okay, this is happening, okay, in what we call rubisco. This is a protein that allows for this to happen. Okay? Now, these 3 molecules of carbon dioxide are going to bind to RUBP. And again, we talked about this. You'll get 6 molecules of 3-phosphoglycerate. It becomes phosphorylated. ATP is used. Okay, 6 molecules of ATP. Okay, and you get 1,3-bis-phosphoglycerate. Okay, that's an intermediate molecule. You can see the phosphorylation on this molecule. That's not going to stick around for very long, especially when NADPH is going to be added. Those inorganic phosphates are going to be removed, and you'll get 6 molecules of glyceraldehyde 3-phosphate. One's going to be donated to glucose, or in some case fructose. Remember, glucose and fructose are the same chemical formula, okay? And that just keeps continuing on and on and on. That molecule gets reconfigured. Okay? ATP is utilized again. So I said at least 7. Okay, it's more like 9 in most cases. 9 molecules of ATP. Okay, in order for the Calvin cycle to turn once. So this ATP is utilized, RUBP is reformed. Okay, so this is a very continuous cycle. Okay, energy is being expended, but remember, energy is being expended to bind up carbon dioxide into a glucose molecule. So we'll come back and we'll talk about this. Okay? We're looking at 9 molecules of ATP per turn of the Calvin cycle. Okay? So, I mean, you're kind of looking at roughly 40, 45, 46, 48. It just depends on the plant, how many molecules of ATP are being utilized. But we could just round it up and just say, look, you're looking at 60 molecules or so of ATP to form 1 molecule of glucose. Okay? I think that's probably right, somewhere in that range, okay? If you round it up. So let's think about that when we move on to the Krebs cycle and cellular respiration, because we'll talk about the breaking down of a glucose molecule. How much energy do we get from breaking down a glucose molecule? We can say that we have to expend, or a plant has to expend, about 60 molecules of ATP. Now remember, that's all free energy as long as photons are available, water is available, and carbon dioxide is available. It's free. Okay? But let's talk later about, well, how much energy does a human get from 1 molecule of glucose? Okay? All right, so we'll approach that at a later date. Okay?
The Calvin cycle is a series of reactions that results in conversion of carbon dioxide into the organic molecules needed to build new cells. It occurs in the stroma, which is the area of the chloroplast surrounding the thylakoid membrane. During the Calvin cycle, carbon dioxide, CO2, is added to a 5-carbon molecule called RUBP. The resulting 6-carbon molecule is unstable and quickly splits into 2 3-carbon molecules called 3-phosphoglycerate. Using energy from ATP and reducing power from NADPH, which are products of the light reactions, the pair of 3-phosphoglycerates move through a series of reactions and are converted into 2 molecules of glyceraldehyde 3-phosphate. When several of these glyceraldehyde 3-phosphate molecules have been produced, some combine to make glucose, while others are reused in the Calvin cycle. To generate an entire new glucose molecule, the cycle has to turn several times, because each turn of the cycle adds only one carbon atom from each molecule of carbon dioxide.
Okay, and we know that several times in this case, in a typical plant's Calvin cycle is 6 turns because it's only adding a single carbon at a time to the glucose molecule. Now, again, I have to reiterate this as much as possible. This is just a typical plant that's been examined. Not all plants work this way. Not all plants' Calvin cycles are the same. Not all plants utilize that much ATP, 9 molecules of ATP. Some use 7, some might use 10, some might use even fewer than that. It just depends on the species of plant, how much energy is needed in order to do this. It also depends on what's going to happen with that molecule. Is glucose being made? Is fructose being made? What's going on? How much further development does that sugar molecule need? Are you then taking these sugar molecules and you're making starches or sucrose, or what's going on? Then more energy is going to be utilized in order to make those more complex molecules at a later date. Okay? All right, now that's the Calvin cycle. So now we've got light independent, forming ATP and NADPH, light dependent, forming ATP and NADPH, light independent, or the Calvin cycle, taking those two energetic molecules, ATP, the currency of the cell, and NADPH, the electron carrier, taking them and converting carbon dioxide into glucose or a carbohydrate. That is photosynthesis. Now, let's talk about things that can disturb photosynthesis. One way that photosynthesis can be disturbed is what we call photorespiration. Now, there's lots of plants that carry out photosynthesis in the C3 photosynthesis way. So exactly what we just described. Photons come in, captured, okay? Water's taken up through the roots. That's breaking an electron off. ATP is formed. NADPH is formed. Those molecules are used in the Calvin cycle. Carbon dioxide comes in. Oxygen leaves. Okay? But the issue is when it's hot out, anytime you open your stomata to take in carbon dioxide for the Calvin cycle and release oxygen as a waste product, water leaves the system. So we need to prevent water loss. Right? And so plants have stomata. We already talked about this, or stoma. They have guard cells that can open and close, right, to prevent water loss, but they have to open up in order to prevent gas exchange. If it's closed, then the stomata cannot have gas exchange. Okay? And again, like I said before, this is why I keep claiming oxygen is not a product of photosynthesis. Oxygen is a waste product of photosynthesis. Because if the plant does not rid itself of oxygen, it will die. Okay? Oxygen is poisonous to the plant, and that's because oxygen binds to rubisco. It binds up, okay, the Calvin cycle, and it stops it from working. Okay? So oxygen will fall in place of CO2, but it disturbs the whole system and it breaks the Calvin cycle. Okay? So you cannot allow oxygen to build up in the cells of plants, otherwise the Calvin cycle, it just, falls apart. It doesn't work any longer. So you have to exchange oxygen for carbon dioxide. Okay? So again, like I said, okay, the enzyme rubisco will fix oxygen instead of carbon, and that causes photorespiration, and it short circuits the Calvin cycle and ends photosynthesis. Okay? So oxygen is not a product of photosynthesis. It's a waste product, and it needs to be expelled from the leaves. Okay? So how do plants do it when it's hot out? Okay? Well, if you open your stomata when it's hot out, you're going to lose a lot of water, okay? Which is not what you want to do, because that water is needed for the electrons. It needs to be, broken down so you have electrons donated to the P680 site. But you can't just close the stomata, because oxygen will build up and it'll short circuit the system. So what we look at is some plants have evolved to be C4 photosynthetic plants. And so instead of doing C3 photosynthesis, they have a little bit of a twist on photosynthesis. And so there's lots of plants like this. Pretty much all your grasses. Okay? So sugarcane and corn, those are grasses. Okay? But Bermuda grass, bluegrass, everything that you can think of from grasses that you would know of. But also a lot of desert plants that are not succulents. Okay, so non-cactus and things like that. But like things like sagebrush and rabbit brush and greasewood and other plants like that, they're all C4 plants because they're adapted to the desert environment. So what have they done? They have the capability of fixing carbon. So instead of opening your stomata and bringing in carbon dioxide each time you want to turn that Calvin cycle, each time you want to produce a carbohydrate, they can open their stomata, sometimes at the night, during the nighttime, sometimes during the daytime when it's cooler, and they can suck in a bunch of carbon dioxide, and they can fix it. They can trap the carbon dioxide in special cells that we call bundle sheath cells. And they fix carbon dioxide in the form of an acid, but then it can be reconverted into something that's useful, and we'll look at that here. Okay, so they can bring it into the mesophyll cells, okay, just like a C3 plant would do. But they bring it in and they can bind it to a phosphoenol pyruvate. Now, pyruvate is a very important molecule when it comes to cellular respiration. When we talk about breaking down of glucose, the first molecule, the first thing that you break down glucose into, so you take a glucose molecule and you break it down, pyruvate is that molecule. So, C4 plants are using that pyruvate to basically bind carbon dioxide to it. Okay, and you're going to get these intermediates, but then you get a malate. Malate is going to be the molecule that gets to donate carbon dioxide to the Calvin cycle. So instead of carbon dioxide just coming straight in and going to the Calvin cycle, okay, you get these intermediate. Okay, and you can store oxalate, acetate. You can store that in the cells. Okay, and you can store malate and you can store pyruvate. Now it takes a little more energy for this to occur. So C4 plants, they still are expending energy down here in the Calvin cycle, but they're also expending energy in order to fix carbon to some of these intermediate molecules. But they can get away with it doing this because now they can conserve water in desert regions, which is highly, highly evolutionarily efficient. Okay? Now, another way you can do that, though, is to not do photosynthesis when hot weather is occurring. So do it at a different time. Right? So another way that photorespiration can be avoided is by store your water. So when you open your stomata, you have water to give up in some cases, but you can store lots of water. So when it's wet, you can store as much as possible. And so cacti and pineapples and other succulent plants do this, okay? But also do what we call crystalline acid metabolism, or CAM plants. So this is kind of like a hybrid between the C4 pathway and the C3 pathway. So they do the C4 pathway at night, so they open their stomata at night, bring in carbon dioxide and fix it, okay? And then they do the C3 pathway during the day when photons are available. So it's kind of the best of both worlds in essence, okay? So they don't have specialized cells in CAM plants. They just do these at different times of the day, okay? And it's a little bit different pathway, but both are much more efficient in arid environments, C4 plants and CAM plants, than C3 plants. Okay, so this is a way that you can get around water loss and the buildup of oxygen in the plant cells. Okay. All right, next thing we're going to do is we're going to move from the construction of carbohydrates, which we just talked about in the Calvin cycle, and then we're going to take that and we're going to put it in an organism that consumed it. It could be the plant that made it, but it also could be an animal that ate the plant, okay? Or an animal that ate the animal that ate the plant, and on and on and on. But we're going to then deconstruct that carbohydrate, that C6H12O6 molecule. We're going to break it down, okay? And that's what we call cellular respiration. So that's next up in the lecture series.